Hello and welcome to our last lecture on Chapter 7 on Section 7.7, where we're going to talk about regulation of cellular respiration. Now, when we go through cellular respiration, we don't want to just keep pumping ATP to create ATP, but instead we need balanced amounts of energy in the form of ATP, and we need to use the resources we have appropriately. So let's go ahead and go through our learning objectives. We want to describe how feedback inhibition would affect the production of an intermediate or product in a pathway, and identify the mechanism that controls the rate of transport of electrons through the electron transport chain. First, we'll start with our regulatory mechanisms, and we've discussed this before, but we have our GLUT proteins, which are our glucose transporter proteins, right? They are going to work to allow that glucose to enter into the cell. And yes, insulin plays a role here. I always call it the key to the door to let glucose in. So insulin has to bind to the receptor first, and then we have many different types of GLUT proteins that are going to be embedded on the plasma membrane that allows glucose to move into the cell. Now we can work to increase or decrease the enzymatic activity by utilizing something called allosteric effectors. If you remember when we use that word allosteric in reference to our lecture on enzymes, remember that it is going to alter the steric structure of our enzyme, and that can affect the configuration of the active site where the substrate is going to attach. And this can increase or decrease the affinity or the attraction of that substrate to bind to the active site. And examples of these allosteric effectors could be ATP, ADP, AMP, NAD, and NADH, which all can change that configuration of the active site. And this is going to help to create a feedback mechanism, meaning that once we see the overall concentration of a chemical decrease, it can diffuse away from the protein and the control becomes relaxed. Now let's talk about control of catabolic pathways. We're going to talk about glycolysis first. And notice how in this first step, when we are taking glucose and utilizing the enzyme hexokinase to phosphorylate it, it becomes our glucose 6-phosphate. Now this phosphate is making this molecule want to stay in the cell. Otherwise, it would just diffuse out and glucose would not continue forward. The other thing that we need to see is this third step over here. This is also going to be a regulatory step in which we are going to have fructose 6-phosphate be converted using an enzyme called phosphofructokinase to add another phosphate group, and we get fructose 1,6-bisphosphate. Now, if we have high levels of ATP, or citrate, and I'm going to go back to show you where citrate is. Here's our Krebs cycle, so not the glycolysis pathway, but in the Krebs cycle, if we see we're getting a lot of citrate or we're seeing a more acidic pH, that will decrease the enzyme's activity. And this is because we realize we don't necessarily maybe need to go into the full pathway of glycolysis because we have enough ATP. We have this Krebs cycle continuing to then go into the electron chain transport and create more ATP. So again, we're going to be resourceful and we're going to hang on to these molecules for when we need them. And the last part of glycolysis we'll talk about as far as a regulatory step is the last step, the 10th step here, where we have PEP being turned into pyruvate utilizing the enzyme pyruvate kinase. And this can catabolize into alanine, the amino acid, if needed as well. And we see that if more energy is needed, then we want to create more pyruvate, and then that gets turned into acetyl-CoA through an enzyme known as pyruvate dehydrogenase. These enzymes can be controlled by either using a enzyme that is a kinase to phosphorylate it, which actually is going to decrease activation, or utilizing the enzyme phosphatase to dephosphorylate it, meaning taking a phosphate off, and that helps to reactivate the enzyme. Now let's talk about control within the citric acid cycle. We're going to have our isocitrate dehydrogenase and our alpha ketoglutarate dehydrogenase serve as two enzymes to make our first NADH. And you could see them at play over here and here. And when we have enough of the NADH and ATP, the reaction rate is going to decrease. Again, we're being conservative, making sure we're not going to make too much. If we have increases in ADP molecules, though, meaning that we don't have a lot of ATP, then our reaction rate is going to increase, so we can make more ATP. And our alpha dehydrogenase is also going to be affected by the levels of succinyl-CoA. And you could see that that is the end product over here for that enzyme. So if we see increased levels in that, then we are going to slow down this process or start to deactivate some of these enzymes so that we're not producing so much of it. Now let's talk about control of electron transport chain. This is really affected by levels of ADP and ATP. When we have greater ATP consumption by a cell, then we start to see a buildup of ADP, and that will signify for us to drive the reactions forward so that we can create more ATP. So as ATP usage decreases, we start to see that ADP concentration is going to decrease and ATP will start to increase inside the cell. This ATP then triggers the cell to slow down the electron transport chain. So once again, we can be conservative. Here is a summary slide from your book. This is table 7.1. It's really great to kind of take a look at these limiting steps or important steps so that we can control cellular respiration.